Okay. So we have done the we have already done the alpha beta T cells. Now we are moving on to the gamma delta T cells. And if you remember in my first class, where in the lymphoid, in this series, in the lymphoids, if we in the hematopoiesis stage, we get the lymphoid progeny and the myeloid progeny. From the lymphoid progeny, we get the T lymphocytes and the B lymphocytes. The T lymphocytes can further differentiate into the alpha beta T cells, the gamma delta T cells, the natural killer T cells, and we get another category of lymphoid progeny, the natural killer cells. So today we'll be taking up the much less known or the less popular T cell, that is the gamma delta T cell. Prior moving to gamma delta T cell, let us have a look at the comparative functional and the anatomical dissection of the naive T cell, the effector T cell, and the memory T cell. Points to be mentioned is that the naive T cell is found only in the secondary lymphoid tissue, while the effector T cells are found on the peripheral tissue. In fact, the effector T cells are also found on the secondary lymphoid tissue. So this is a point to be mentioned. Second point that is important, I want you all to highlight, is that in case of a naive T cell, the activation of the T cell requires a time span of as long as 20 to 30 hours. But if you look into the activation time required for the effector T cell and the memory T cell, it's almost 1 hour. So you can see that in case of the primary immune response, the adaptive immunity is roped in almost after 30 hours. While in case of a secondary immune response, the adaptive immunity is quick to respond. Now comes to the lifespan. Just if you look, the lifespan of a naive T cell is 5 to 7 weeks, while that of a effector T cell is 2 to 3 days, and again that of a memory T cell is up to 50 years. So memory T cells live long. Coming to the category of cells which are going to enter today, but before entering into the gamma delta T cells, a brief comparison between the one we have already studied, the more popularly known alpha beta T cells. If we look into the comparative features of the two cells, I'd like to highlight on specific points. You can see they are part of the induced innate immunity, and the alpha beta T cells are part of the adaptive immunity. So the statement that T lymphocytes only play a role in adaptive immunity is grossly erroneous, because as you can see from this picture itself, the the gamma delta T cells do play a role in the induced innate immunity. Next, if we look into the TCR, it is made up of the gamma delta chain. They are made up of the alpha beta chain. If we look into the distribution, the gamma delta T cells are present in the epithelial layers, skin and mucosa. And when we did the structure of salt, if you remember, that the gamma delta cells are present in the mucosal layer as intraepithelial lymphocytes. In case of the alpha beta T cells, they're found only in the primary and the secondary lymphoid organs. And they also, to some extent, when there is activation of the immune system, we do observe the presence of alpha beta T cells in the peripheral tissues in the skin also, if you remember the crypto patches. Now coming to the presentation, antigen presentation, always the alpha beta T cells recognize their antigen complexed with major histocompatibility complex, but the gamma delta T cells can recognize antigen. They do not need the MAC processing of the antigens. They can recognize their antigen by when they're complexed with the non-polymorphic CD1 complex. And this particular category actually tells, this particular property of category actually tells us the fact that the gamma delta T cells have evolved earlier than the alpha beta T cells, as the gamma delta T cells do not require the antigen to be processed in a defined manner as required by the alpha beta T cells. So the gamma delta T cells recognizes the antigen in a manner very similar to the recognition done by the pattern recognition receptors. Another important thing I'd like to highlight is that if you look into the different populations that are produced by both the category of cells, you can see for gamma delta, We are getting helper 1, helper 2, helper 17 cytotoxic T lymphocyte activation. In case of alpha beta also, we get these populations, and regulatory T cells are observed in both cases. Coming to the various antigens that can bind to the gamma delta T cells, as you can see in the picture. But before coming and getting into the details of the picture, I just want to give you 2 to 3 important informations regarding the gamma delta T cells. The first information was that I said that the gamma delta T cells evolved prior to the alpha beta T cells, since the gamma delta TCR recognizes ligands in a way similar to the ligand recognition system of the pattern recognition receptors. Second, as I said, the gamma delta T cells do not require the involvement of the conventional MHC nor the processing and presentation of antigens by antigen presenting cells. Intact proteins or peptides from pathogens or stressed host cells and non-protein antigens such as phosphorylated nucleotides can serve as gamma delta T cell ligands. So you can see the variety of molecules that can serve as ligands for the gamma delta T cells. Third important point, once activated, the gamma delta T cells respond very similarly like the alpha beta category by proliferating and differentiating into gamma delta T helper cells and cytotoxic T lymphocyte effector cells. So they produce this category of population. Finally, like macrophages, natural killer cells and natural killer T cells, gamma delta T cells can be counted among the sentinels of induced innate immunity. Sentinels means bodyguard. So you can well assume that the gamma delta T cells play a greater role in innate immunity rather than taking part in the adaptive immunity. I further said while explaining the differences that if you look into the anatomical distribution, the gamma delta T cells are dispersed among the epithelial cells and of the mucous membranes and constitute one type of specific cells known as intraepithelial lymphocytes. When these intraepithelial lymphocytes are found in the gut, specifically in the gut, they are called as small I, intestinal intraepithelial cells. So the small I symbolizes intestinal. So small I, capital I, capital E, capital L. That's how they are named then. Another important thing, when the intraepithelial lymphocytes are resident in the top layers of the skin, they are sometimes called as dendritic epidermal T cells. The and the abbreviation for that is capital D, capital E, capital T, capital C. It's DETC. The location of the gamma delta T cells at the mucosal surface allows them to be among the first to confront invading pathogens or injurious substances. So as I again said, they are playing a great important role in induced immunity. Regarding the antigen presentation function, I said that the gamma delta T cells appear to bind to non-polymorphic CD1 family of MHC-like molecules that present non-peptide antigens to alpha beta T cells. So it's the same non-polymorphic CD1 molecules which is actually presenting the antigen to the gamma delta T cells, which present peptide antigen to the alpha beta T cells. It's the same CD1 molecule. Coming to the picture, this picture, if you look into this picture, this is basically alpha beta T cell presentation. As you can see, we have the alpha beta receptor, this is a CD8 molecule, MHC molecule, and the antigen. These three pictures are of the gamma delta cells. So as you can see, the gamma delta cells can recognize various category of antigens. In the A category, the first picture, as you can see, the necrotic epithelial cells leak stress protein. The necrotic epithelial cells leak stress protein, which such as HSP78, which are recognized by the gamma delta, which are recognized by the gamma delta T cells. Then, as you can see, the stressed epithelial cells are producing a category of molecules. The MHC, it's very similar to the MHC category of molecules. So the stressed epithelial cells produce this category of molecules, and the recognition is again done by the gamma delta T cells. Now moving on to the third category, the activated macrophages, the B lymphocytes, the dendritic cells, and the monocytes. So these category produce antigens complexed with the CD1 molecule, the non-polymorphic MHC, and the gamma delta T cells recognize them. So these are the variety of antigens that can be recognized by the gamma delta T cells, and accordingly the gamma delta T cells are activated. This is a very simplified diagram of the development of the gamma delta T cells. It's the development process of the gamma delta T cells. So as you can see, initially the gamma delta T cells arise from the same natural killer T lymphocyte precursor in the bone marrow that produces the alpha beta T cells and the NK cells. So they arise from the same precursor. Now what happens? Some of the gamma delta T cells migrate from the bone marrow into the mesenteric lymph nodes, fetal liver, and intestinal cryptopatches. So these gamma delta T cells are known as extrathymic gamma delta T cells. But some molecules, or natural killer T lymphocyte precursor migrate into the thymus. Here, the precursors migrate into the thymus and receive IL-7. The IL-7 signaling actually gives rise to the double negative 3 stage. So the double negative 3 stage is achieved when the initial double negative 1 cell receives the interleukin 2/7 signaling. If the double negative 3 cells express high level of the interleukin 7 receptor alpha, that is, they are receiving strong signals from interleukin 7, receive strong interleukin 7 signaling. It is these cells that only mature to the gamma delta T cells. So very simply, in a very simple manner, if I say double negative cells receiving the complete signaling of interleukin 7 matures to the gamma delta T cells, while the double negative cells which do not receive the IL-7 signaling to its highest or the fullest extent is known as matures to the alpha beta T cells. So that was a simplified differentiation pathway between the alpha beta and the gamma delta T cells. So the interleukin 7 actually acts as the pivotal molecule, which its presence indicates that the double negative stage will become the gamma delta T cells. The absence of this signaling indicates that the double negative 3 cells will become the alpha beta T cells. Now, before moving on to that, let me come to this picture. So this picture details the functional capacity of the gamma delta cells. As you can see, once the gamma delta cells are activated, this is activated gamma delta cells, they can generate gamma delta helper 1 cell, they can generate gamma delta cytotoxic T lymphocytes, gamma delta helper 2 cells. The gamma delta cytotoxic T lymphocytes and the T helper cells of either category has a huge immunoregulatory function and a control over the alpha beta T cells. In case of the gamma delta T cells, it can mediate cytotoxicity in the very similar manner which we discussed for the alpha beta T cells. In fact, it can also kill an alpha beta T cell if the alpha beta T cell is exhausted or if the alpha beta T cell is hyperactivated. So it can kill an alpha beta T cell. It can kill a bacteria by direct liberation of granulysin, which is a microbicidal, which has a microbicidal activity. Coming to these two categories, as you can see, the gamma delta T cell can differentiate into helper 1 and helper 2 category. Now, concentrating on helper 1, it can produce a host of cytokines, again regulating the immune system in its own level. It can produce interleukin 2F and gamma to activate a B lymphocyte, bringing in about the isotype switching of B lymphocytes. It can produce, it can differentiate a helper, alpha beta helper cell into a helper 1 category or a helper 2 category, depending on the cytokines that are being produced. It can activate a macrophage. Okay. Another thing, just look into this, that if the helper 1 category of gamma delta cells are activated by the presence of this pro-inflammatory cytokine, it can in fact inactivate a neutrophil. So if there is an extreme amount of pro-inflammation going, and if the gamma delta helper 1 category are activated, at times they do inhibit the function of the neutrophils, as you can see here, and they also produce keratinocyte growth factors in the skin, and they can activate the gut epithelium. I mean to say, they help in proliferation of the keratinocytes. Not only that, they also activate certain keratinocytes, inducing them to produce certain pro-inflammatory cytokines. So this is the host of function or the host of activity that a gamma delta cell actually provides. Now, coming to another category of cells, the natural killer T cells. So, as you can see, when we talked about the gamma delta cell, there is this mother cell. From this mother cell, some moved onto the peripheral tissues, while some moved into the thymus, which developed into the double negative 3 stage. From the double negative 3 stage, we are seeing two populations, one the alpha beta and the gamma delta. So I'm again coming back to this particular cell. So we have the NKT cell. So this is the maternal cell. So this is the, now what happens, if we compare the picture, we are seeing the double negative cells, and the double negative cells through 4 stages ultimately get converted to double positive alpha beta T cells. So keep in mind that the natural killer T lymphocytes are basically a variant of the alpha beta T cells. How do they differ? During the process of positive selection, the alpha beta T cells are selected for either the CD4 receptor or the CD8 receptor. But some of the alpha beta T cells might bind with the non-polymorphic CD1 receptor. So those which binds with the CD1D receptor get selected and behave as the natural killer T lymphocytes. So don't confuse for the fact that we get alpha beta and gamma delta from the double negative 3 stage. We get the alpha beta T cells and the natural killer T cells from the double positive T cells. Now talking about the natural killer T cells, the natural killer T cells are of lymphoid lineage, and they share physical and functional characteristics of NK cells and T cells. They are low in numbers and are found virtually everywhere. NK cells carry what is sometimes referred to as the semi-invariant alpha beta T cell receptor. Human natural killer cells express both the killer inhibitory receptor, and the KIRs also. So these inhibitory receptors are properties of the natural killer cells, which we'll take up on just later after this. And human natural killer T cells also express the activatory NK receptor NKG2D. T cell co-receptors. The human natural killer T cell population includes both CD4 positive and CD8 positive cells, as well as CD4 negative and CD8 negative cells, but that is a very rare variant, because I said that actually the selection happens after the double positive cells have developed. Regarding the antigen recognition and activation, it has been seen that the TCRs of the natural killer T cell appear to recognize glycolipid or lipid structures present on the non-polymorphic CD1D molecule. And if you remember the CD1D, it has a structural similarity to MHC class 1, but behaves more like MHC class 2. So the presentation of the antigen is done via the exogenous pathway. In addition to MHC class 2, CD1D is also expressed by professional APCs. Another thing I'd like to point out when I mention this point is that professional APCs by default always usually express the CD1D molecule, but MHC class 2 expression is a matter of choice if there is a presence of antigen. Another important thing we need to know about the natural killer T cells is that in addition to the TCR ligation, the NKT cells can be sometimes activated by the natural killer activatory receptors. So this was about the NKT cells. Coming to the activation and the effector functions, as you can see from the picture, the NKT cells are activated by interaction of their variant alpha beta T cell receptors with the CD1D ligand presented by the antigen presenting cells, and then they become activated natural killer T cells. Natural killer T cells, once when this signaling crosstalk has been completed, also exhibits the CD40 molecule, CD40 ligand. So they express the CD40 ligand by which they can interact with the dendritic cells. So the CD40 ligand interaction occurs, and as you can see, the dendritic cells gets activated, thereby activating the alpha beta helper 1 population. And the natural killer cells, the activated natural killer cells, can also produce a host of cytokines that can lead to cytotoxic killer lymphocyte activation, that can lead to helper 2 differentiation, and also a number of cytokines that can lead to the activation of natural killer cells. So this is basically the differential function of the natural killer T cells. Now coming to the last category of cells, which are important and which was not there before, are the natural killer cells themselves.